Punnett Squares.
Punnett Squares. The implications of Mendel's work are made easily visible by the work done by Reginald Punnett in the early 1900s. He extended Mendel's work and developed a schematic approach to predicting the outcomes of breeding. The probabilities of the genotypes and phenotypes of offspring resulting from the breeding of parents, given their genotypes, is simple to calculate for single traits.
Set up a Punnett square by determining the alleles that could be contributed by each parent. Every parent has 2 alleles for each gene, one on each of its homologous chromosomes. During meiosis, homologous chromosomes are distributed into different gametes, so each allele is in a different gamete. For a gene that has alleles capital A or lowercase a parent could contribute one of these combinations, both uppercase A's, a mix of uppercase and lowercase, or 2 lowercase a's. A Punnett square is divided into 4 equal-sized smaller squares as shown here. The alleles of one parent, so 2 letters, are written on top and 2 letters for the alleles of the other parent along the side. For example, write the alleles for a true breeding plant with purple flowers on top and for a true breeding plant with white flowers down the side. So we have 2 capital P's on top and 2 lowercase p's on the side. Now fill in each square with 1 letter from above the box and 1 letter from the side. We usually write the capital letter first. We'll show you the first box and you do the rest. That'll be shown on the next slide. So this box has a capital P and a lowercase p. Each square represents a 25% chance of that genotype being the genotype of the resulting offspring. Since capital P is dominant, what will be the distribution of flower colors of these offspring? This is the F1 generation. All of these offspring will have purple flowers, since capital P is dominant and appears in each box. The only way to get a white flowering plant is for capital P to be absent. Now, let's crossbreed 2 of the offspring from the previous page together using a Punnett square to get the F2 generation. In this case, both parents are capital P, lowercase p. So capital P, lowercase p, capital P, lowercase p. Fill in the square and check your answer on the next page. So this will be 2 capital P's. Here we have an uppercase P and a lowercase p. Here we have an uppercase P and a lowercase p. And here we have 2 lowercase p's. This is the first step. Now fill in the boxes. All right, I'll do this again. 2 capital P's, a capital P and a lowercase p, a capital P and a lowercase p, and then 2 lowercase p's. What will be the probability of each possible genotype in the offspring? The genotypes in the 4 boxes are 2 capital P's, a capital P and a lowercase p, a capital P and a lowercase p, and 2 lowercase p's. Each appearance represents a 25% chance of that genotype occurring in the offspring. So the probabilities are a 25% chance of 2 capital P's, a 50% chance of a capital P and a lowercase p, and a 25% chance of 2 lowercase p's. What will be the probability of each phenotype in the offspring? In other words, what is the likelihood of the offspring having purple or white flowers? Anytime capital P is in the genotype, the plant's flowers will be purple. That means that there is a 75% probability of offspring having purple flowers and a 25% chance of offspring having white flowers, because here, all of these and these will have purple flowers and only the 2 lowercase p's will have white flowers. The only genotype that leads to white flowers is this one with 2 lowercase p's in the lower right corner square. In the flower color example, 2 uppercase p's and an uppercase lowercase p plants have the same phenotype but different genotypes. So the genotypes are 2 uppercase p's, an uppercase and a lowercase p, or a lowercase and an uppercase p. A phenotype is the observable characteristic, purple flower. A genotype is the organism's set of genes. The above three genotypes result in purple flowers, since capital P is dominant. In this flower color example, a lowercase p plant has white flowers, so both p's are lowercase. This is the only genotype which results in white flowers. That is because lowercase p is for white flowers and is recessive. Use the same steps to solve this problem. The answer is on the next slide. Tall is dominant to short. If you cross a homozygous tall pea plant with a heterozygous tall pea plant, what percentage of the offspring will be tall? Alright, so a homozygous tall would be 2 capital T's, and then a heterozygous would be one of each, and it would be tall, with 1 capital T and 1 lowercase t. So what percentage of the offspring will be tall? Well, let's go through. This has 2 uppercase T's. This also has 2 uppercase T's. This has 1 uppercase, 1 lowercase, 1 uppercase, 1 lowercase. Now, all 4 of these options have a capital T, at least 1 capital T, which means 100% of these offspring will be tall. So here we have 50% homozygous dominant, so 2 capital T's, and we have 50% heterozygous, so mixed, 1 uppercase, 1 lowercase. 100% tall is the phenotype because the only way we would get a short pea plant would be having 2 lowercase t's, which does not exist here. Use the same steps to solve this problem. The answer is on the next slide. Yellow seed is dominant to green seed. If a heterozygous yellow seed pea plant, so that would be one of both, so uppercase Y and lowercase y, is crossed with another heterozygous yellow seed plant, so uppercase Y, lowercase y, what percentage of the offspring will have yellow seeds? Alright, so here we have 2 capital Y's, so that's definitely yellow. Here we have uppercase and lowercase, so that's also going to be yellow. Uppercase, lowercase, that will also be yellow. However, here we have 2 lowercase y's, so this one will be green. So what percentage of the offspring will have yellow seeds? 75%. Alright, so we have 25% homozygous, which is this one, 50% heterozygous, which are this one, and then 25% homozygous recessive, which is that guy. The phenotype distribution would be 75% yellow, 25% green.
Test cross. A test cross involves breeding the individual whose genotype we are trying to determine with a homozygous recessive individual. We know the genotype of a homozygous recessive individual. In this case, white is recessive to purple, so if we have a white individual, it must be little p. If any offspring display the recessive phenotype, then the parent must be heterozygous. So in this case, if we have white flowers, when we cross a white plant with a purple plant of an unknown genotype, that unknown genotype must be heterozygous, big P, little p, because these white flowers had to have received a little p from both parents, including the purple plant with unknown genotype. Now you may ask, well, what if these 2 flowers, the white and the heterozygous purple, only had a couple offspring, and all of the offspring just happened to come out purple? This could happen, but it's unlikely with the more offspring that they have. If they were to have 50 offspring, for example, it would be statistically impossible that all of those flowers would come out heterozygous purple and none of them homozygous white. Dominant alleles. Just because an allele is dominant does not mean that it is more common than its recessive counterpart in a population. For example, 1 out of 400 babies in the United States is born with extra fingers or toes, polydactylism. The allele for this unusual trait is dominant to the allele for the more common trait of 5 digits per appendage. In this example, the recessive allele is far more prevalent than the dominant allele in a population.